This is the brain of a cat. You can see the cells in this photomicrograph of the cerebral cortex. The cell bodies are micrometer, much smaller in diameter. The uninsulated tip of a microelectrode is usually about 10 to 20 micrometers long and about 1 micrometer wide. To contact one particular neuron alone, the electrode tip must be held in place close to the cell or its fiber. Stereotaxic instruments, like this one, have been designed to provide this stability. The animal's head is fixed in 3 ways, shown on this cat skull. Bars are inserted into the ears. Other bars press down on the ridges of bone under the eyes, and a bar pushes up on the roof of the mouth. This instrument not only keeps the head rigid, but also makes it possible to insert an electrode to a known point deep in the brain. Atlases have been made that define the position of each part of the brain in terms of coordinates, based on the line between the ear bars and eye bars. So the position and angle of the electrode can be arranged by reference to these coordinates to explore any particular area. The animal has to be carefully prepared for the experiment. After the cat has been anesthetized and its head is fixed in the stereotaxic instrument, we have to make a small hole in the skull. First, an incision must be made in the scalp, and the skin reflected. Then, a tiny hole is drilled in the correct position. In this case, the hole is directly over the visual cortex in the occipital lobe of the left cerebral hemisphere. Between the skull and the cortex is a tough membrane called the dura, and this must be removed. When this has been done, the cortex is revealed underneath. Now the cat is in position on the experimental table where the recording takes place. The experiment can last for several hours or even days, and careful attention must be paid to the cat's physical condition. It's kept warm by a thermostatically controlled blanket, and if necessary, it is artificially respirated with a pump connected to a cannula in the trachea. Anesthetic and sometimes paralyzing drugs are intravenously. To check on all these systems, we are monitoring the cat's temperature, its electrocardiogram, and its electroencephalogram. Now we're ready to begin the experiment. The microelectrode is in position, and I can advance it into the brain by means of a hydraulic system. The tiny electrical signals picked up by the electrode are led off through a cable to an amplifier which boosts them. These amplified signals are displayed on an oscilloscope and played through an audio monitor so that we can hear what's going on. As the electrode goes down through the cortex, you can hear and see the noise of the background cortical activity. If I advance the electrode very slowly, it should be possible to isolate impulses from just one neuron. Each click that you can hear is one action potential. Look at the impulses on the oscilloscope. The waveform of the impulse is virtually identical every time, which is a good indication that the electrode is picking up from just one neuron and not from several. Now, if the time base of the oscilloscope is slowed down, you can see several impulses on each sweep. This is the spontaneous firing of a single nerve cell. It is recorded with an extracellular microelectrode while the cat is held firmly in the stereotaxic apparatus.